What we have discovered is that breast cancer is a constellation of 10 different diseases. The way we did this was by profiling these tumors at the molecular level, both DNA and RNA. And this is a classification that is what we call a driver-based classification, because it's based on the pattern of oncogenes and tumor suppressor genes of the 10 different subtypes that we have identified. And it's going to be very important in future to take into account this taxonomy precisely because it's based on drivers. And so therefore, these 10 different diseases are going to require different treatment and different management. And the way we're going to bring it to the clinic is going to have to be to produce a test that can be used in routine pathology laboratories to classify the tumors into one of these 10 subtypes. One of the areas that we have learned is that the concept of cancer stem cells is not quite what we first conceptualized, if you want, 3 or 4 years ago. We realized that there is not going to be a universal cancer stem cell population. We realized that there is not going to be a set of markers that will identify cancer stem cells across all breast cancers, for example. And we also increasingly recognize that there is plasticity in tumors. In other words, there are cells that can at one moment behave as stem cells and the next moment they don't behave as stem cells. So I think much more important than the concept now of cancer stem cells is the concept of cancer cell plasticity, the fact that there is clonal evolution, there is functional evolution within tumors, and I think that that's much more the direction in which we're going. So I think 10 years from now, we will have a dramatic difference in the way we're going to be managing breast cancer compared with what we do today. On one hand, we're going to be stratifying patients into these subclasses that we now recognize. Breast cancer is not one disease, it's 10 diseases, and it's going to be very important that when the patient is first diagnosed, you assign them to one of these 10 subtypes, because as I said, you're going to be managing them differently. So I think the other thing that we're going to see is an increasing use of liquid biopsies. This is using blood samples to track both circulating tumor cells, but increasingly cell-free DNA that comes from tumor cells, which we designate as circulating tumor DNA. And the circulating tumor DNA, I think, will have 3 types of use. One will be molecular stratification. In other words, you use the blood as a liquid biopsy to identify, for example, an actionable mutation in EGFR in a patient with a mass in the chest that you identify on a chest X-ray. You identify an EGFR mutation in plasma, and you can just go ahead and treat with an EGFR inhibitor. So that's molecular stratification. The second use is going to be tumor monitoring. So the quantity of circulating tumor DNA reflects the tumor burden, and so it could be used to more precisely determine whether the tumor is responding to treatment or is progressing, and it can give us an indication of response or resistance much ahead, for example, of imaging. And then the third area is going to be to identify mechanisms of resistance and to identify mutations that are associated with resistance to targeted therapies, but also to chemotherapy.